COVID-19 is an infectious disease caused by a newly discovered strain of coronavirus. COVID-19 is a type of virus known to cause respiratory infection in human and also animal. We have seen in past 1 week a rise in active cases and daily number of COVID-19 cases. Almost all the cases were from Sabah and Kedah. Majority of infections seen in Peninsular Malaysia were contributed by Malaysians returning from Sabah. Symptoms of coronavirus can be divided into 3 groups. First one is most common, second one is less common, and third one is serious symptom. Most common is fever, dry cough and tiredness. Less common is sore throat, diarrhea, headache, loss of taste and smell. And the last one is serious symptom, which is difficulty in breathing, chest pain, and also loss of speech.
As stated by the Center for Disease Prevention and Control, nasopharyngeal specimen and oropharyngeal specimen collection are considered acceptable alternatives. Nasopharyngeal specimen collection is usually recommended. The specimen should be collected by qualified technicians who have received biosafety training and are equipped with the corresponding laboratory skill. It is essential to follow proper infection control measures when samples are collected from patients with a suspected coronavirus infection. Personal protective equipment requirements for sampling personnel are N95 mask, goggles, protective gown, and double-layer latex glove. Let's begin with the nasopharyngeal swab. Sample collection should only be completed using sterile swabs made from artificial cotton with flexible plastic shaft. Calcium argonate swabs or swabs with wooden shaft must not be used. These materials can inactivate viral particles or inhibit PCR tests. Furthermore, wooden shafts are more likely to cause patient injury. Single synthetic fiber swab with a plastic shaft is gently inserted through the nostril to the posterior nasopharynx, where it is left for several seconds to absorb secretion before slowly removing while rotating. The swab should be placed immediately into sterile transport tubes containing 2 to 3 ml of viral transport media. If a nasopharyngeal swab cannot be collected, an oropharyngeal sample can also be collected through the mouth. Gently insert the swab into the oral cavity without touching the lips, teeth, tongue, hard palate, or soft palate. This surface should be avoided as they are often colonized by a wide range of microbes. Collect the sample from the back wall of the throat. The oropharyngeal swab can also be placed in the same tube together with the nasopharyngeal swab to increase the viral load. The sample tube should be sealed properly and packed in 3 layers of biohazard plastic bags. The sample should be transported to laboratory as soon as possible after collected. At the lab, there are several tests that can be done to detect COVID-19, such as antigen tests and antibody tests. In Malaysia, there are 2 tests: rapid test kit SARS-CoV-19 antigen as a screening test and real-time polymerase chain reaction as confirmation tests.
Rapid test kit COVID-19 antigen. It is a rapid chromatographic immunoassay for the qualitative detection of specific antigen of SARS-CoV-19. It provides only an initial screening test result, thus the result of this test should not be the sole basis for confirming or excluding cases alone. Inside the kit, we have test device, sterile swab, extraction buffer tube, buffer stand, nozzle cap, and instruction to use. Before we proceed to run the sample, it is advisable to always read instruction for using the COVID-19 antigen kit. Check the expiration date of the back of the foil pouch. Do not use if expiry date has passed. Check the test device and the desiccant pack of the pouch. Yellow for valid, and if it turns to green, the kit will be invalid. Soft reminder to wear full PPE, switch on the biosafety cabinet, and clean the interior surface before the test is carried out. Label all the device kit and buffer with the correct patient information. Insert the swab into an extraction buffer tube. While squeezing the buffer tube, stir the swab more than 5 times. Remove the swab while squeezing the sides of the tube to extract the liquid from the swab. Press the nozzle cap tightly into the tube. Apply 3 drops of extracted specimen to the specimen well of the test device, with the result in 15 to 30 minutes. Interpretation of the test result. Control line band will appear to show that the test is working properly. Test line of SARS-CoV-19 antigen will appear if it's positive. If both control line and test line not appear, or only test line appear but no control line appear, it is invalid. At the end of the test procedure, there are some limitations that we should know. The test procedure, precautions, and interpretation must be followed strictly. A negative result may occur if the level of extracted antigen in specimen is below the sensitivity of the test, or a poor quality specimen is obtained. A negative result may occur also if the concentration of antigen in a specimen is below detection limit of the test, or if the specimen is collected or transported improperly. Therefore, negative result does not eliminate the possibility of COVID-19 infection and should be confirmed by confirmation test. Positive results do not rule out co-infections with other pathogens. Children tend to shed virus for longer periods of time than adults, which may result in differences in sensitivity between adults and children.
A strong laboratory infrastructure is crucial in fighting COVID-19. Some may not notice that the majority of medical decisions involve laboratory information. PCR is the standard method of coronavirus testing, which is known as a method used to make millions of copies of a specific DNA fragment rapidly. However, coronavirus is a single-stranded RNA genome. SARS-CoV-2 genome contains genes that carry the direction for making proteins that are needed to replicate the virus inside the human cell. Now, bear in mind that the aim of this COVID-19 testing is to identify part of the viral genome in the patient sample. The N gene, which carries directions for making the nucleocapsid protein, which is within the virus envelope. This test requires taking a sample from the patient, usually using nasopharyngeal or oropharyngeal swab or saliva sample that placed in the viral transport media. RT-PCR, or reverse transcription polymerase chain reaction, amplifies many copies of a segment of the N gene, since the viral RNA is not enough to be detected in the patient sample. Firstly, primers, a short single-stranded pieces of DNA, recognize unique RNA sequences within the viral genome that bracket the target region of the N gene. Once the binding took place, here an enzyme called reverse transcriptase extends a single-stranded DNA copy of the viral DNA, which also known as complementary DNA or cDNA. The RNA is removed, then the second primer binds to the other side of the single-stranded DNA. After that, take DNA polymerase, play it rolls by extending a second strand to produce a double-stranded DNA copy of the target region of the viral DNA. Cycle 1. This is the part where amplification happening. where this DNA copy undergoes successive cycles, which include few processes. Firstly, denaturation, while the DNA separates into single strands. Second, annealing, both primers bind to the target sequences. And thirdly, synthesize, the polymerase extends a new DNA strand. Cycle 2, the cycle go on, and as well as the cycle 3. So, each cycle, it can be seen that the number of copies of the target region of the viral genome are doubles. Cycle 3, the process is complete. After going through the 30 cycles, there will be up to 1 billion DNA copies of the viral RNA are produced by PCR. In addition, a fluorescent probe is added to allow the detection of the amount of target DNA in real time and quantified after each cycle of PCR. For example, in this graph shows that the detection of 220 in only 2 virus RNA molecules and a negative result in a controlled study. So, as you can see that the identification of a disease is mostly done in the lab. It is undeniable how crucial the role of the lab scientist or MLT in diagnosis of disease, which especially in this particular case, who runs a sample and then interprets the result to ascertain if it's really COVID-19 or not.
Next, let's look at the precautions that we can follow to prevent the spread of COVID-19. First of all, we should maintain social distancing. Stay at least 6 feet from other people. Then, cover your nose and mouth with tissue while coughing and sneezing. This prevents the germs from spreading. Wearing a mask is compulsory when you are in public. Avoid touching your eyes, nose and mouth. After that, stay home when you are sick. Immediately seek for a medical advice. At last, clean your hands oftenly. Use soap or alcohol-based hand rub. Medical laboratory technologies plays an essential role in the confirmation of COVID-19 diagnosis. They perform several testing such as quantitative polymerase chain reaction, serological testing for antibodies against COVID-19, biochemical testing such as monitoring the change in blood cells, blood gas, liver function tests, and also determining the genome sequencing of virus. Medical laboratory technologies is a patient's first hero, a doctor's backbone, and the unseen life savior. Hoping for a COVID-free country. Stay safe everyone. Thank you for watching our video.